soil diseases, which is an increasing problem due to monocrops of certain horticultural crops, due to intensification. In other areas, it's related to production systems and market trends and production schedules, which condition the optimum rotation of crops. It's particularly taking place because of greenhouses due to climate conditions related to nematodes or soil fungi. Now we're running a bit late at the moment, so I will just say that in the future we will have a land availability problem or of a land conditioning problem due to plant protection issues or fatigue in the soil. So integrated pest management will be increasingly important for the health of our soil, and we have 4 experts on this here with us, and they are going to explain everything about soil management and IPM. So as I say, we haven't got much time. So without further ado, I ask the speakers to take their seats on the front row and we will get going.
El primer ponente que tenemos es Francisco Javier Sorribas.
The first speaker we have is Francisco Javier Sorribas, who is a lecturer in plant protection at the Polytechnic University of Catalonia since 1988. He has worked on plant parasitic nematodes, and he's still working on matters related to management of these nematodes and establishing tools to manage our soil naturally. You have 20 minutes. Thank you. I'm going to talk about plant parasitic nematodes, the problems and decision-making tools, and how we can use these tools to design strategies that will enable us to control the nematode in a sustainable manner. Plant parasitic nematodes don't have internal temperature regulation. They're parasites of plants then, and they can establish a relationship from within the plant tissue, establishing an infection, and then they'll stay there for the rest of their lives. Those are parasites. They can migrate to other parts of the plant, or they can feed on the outside of the plant. The main nematodes that cause losses in economically important crops are sedentary endoparasites. The production losses that they cause are variable according to the type of plant, the crop time of year, but in lettuce you see it's about maximum yield loss of 36%, and almost 90% in cucumber. Nematodes are an emerging problem fundamentally because of the prohibition of fumigant nematicides, chemical nematicides, which had been used to control them usually, and also because of intense farming and the environmental conditions in which they develop. So production losses may be greater when there are synergistic relationships with other soil-borne pathogens. The main genus is Melodigene spp, and more than 15 have been described, but this is the one that has most importance. It's characterized by causing these knots in the plants that it attacks, which leads to less growth, lack of nutrition, marks because of insulation, and it can finally kill the plant. As we've heard this morning in various presentations, it's vital to know the biology and the ecology of the agent that you need to control to know how you can combat it through its biological cycle and the cultural cycle. The inoculum is found in the soil either as an egg inside the egg mass that preserve it from adverse conditions, or as a young that comes out. It establishes infection, transforms cells, and then there are 3 successive changes until it reaches its adult stage. And sex is influenced by environmental conditions. It's female when conditions are favorable and male when conditions are unfavorable. And that's an element that can be used to control populations. They have parthenogenetic reproduction, so the females don't need the males, and they can lay from 200 to 1,000 eggs. So populations increase exponentially. Now, looking at the population increase according to population densities when a plant is planted, there is a time when there's a proportionality, which is known as A, can be seen here. When there isn't interspecific combination, population increase is proportional, and there are competition phenomena, and it starts to grow to a balance point known as E. Now, these parameters allow us to catalog plants according to their status as a host. Susceptible plants have high values of multiple rate of multiplication and balanced density, and those that are resistant or poor have low values in these parameters. Resistant plants are species that have specific resistance genes, whereas poor hosts are an intrinsic species of the plant. Now, this allows us to relate this to the multiplication rate and contrast and compare behavior of nematodes in differing plants, or when we use different management systems to compare these to be able to decide in the long term how this can influence on nematode management. I'll come back to that later. A and E values depend on the plant and the number of variations or generations that the nematode goes through during the cultivation. We have tomato, cucumber, and courgette, and watermelon, and these have been charted onto the graph in relation to the values that I've just mentioned. In a continuous line, unbroken line, you see tomato, cucumber, and courgette, and which is more problematic. And then the broken line is watermelon and resistant tomato, where there's low rates of multiplication and a low rate of balance also. How do we know how many numbers of generations the nematode goes through? The rate of development is conditioned to environmental temperature, so we can determine the optimum temperature and the maximum temperature in different crops. What we have observed is that in crops that behave as susceptible hosts to nematodes, there are no differences according to the thermal requisites that the nematode needs to develop. But there are these differences in poor hosts like watermelon, where there are differences according to temperature. Now, this information allows us to use trap crops, for example, green manure, and other products. In normal conditions, in our latitude, the nematode can reach 2 or 3 generations in spring crops or 1 or none in autumn crops. Another important issue is the relationship between the nematode population density and the crop production. This tells us how much crop loss there will be according to the population just before planting. This tells us a T factor, which is tolerance, density below. The density below this doesn't lead to crop loss. And another parameter is M, which is maximum relative production to maximum density of the nematode. Now, according to these values, we can classify the plant as either tolerant when T and M values are high, moderate when they're moderate, or intolerant when the T values are low and the M value is low also. Okay? So they won't tolerate the nematode and there'll be major production losses. Here you can see tolerance in certain crops, in this case in some that were previously susceptible, I beg your pardon, poor, correct speaker, like watermelon, or susceptible like courgette, or resistant like the resistant tomato. So here you have the M value is high and T is also. Whereas with moderately or intolerant varieties, we have susceptible tomato and cucumber, which would suffer very high losses, particularly in the case of cucumber. What happens to the nematode when there are no plants? The young nematodes that are in the soil will survive according to the reserves, the accumulation of reserves they have, and they are used up according to the soil. So if there are reserves, the nematodes population will grow, and these models allow us to predict the growth in population without a plant host. With all of these tools, we can see the influence that different control methods will have at specific different times in management of the plot. When there are when the plot is fallow, when it's just been planted, or methods that operate through the plant. Those that act in the soil, so for example, removal of roots, solarization, black fallow, and so on. What we see initially is that production of eggs in a tomato crop planted from April to September is around 9 million eggs. If we leave that in the soil, it'll be used for the next population. If we remove the roots, we're taking around 9 million individuals per plant. Now, the young individuals that are in the soil seeking a plant will stay there, but solarization and biofumigation, which increase the temperature in a short period of time, will reduce the survival of these nematodes more quickly. With regard to plant actions, such as green manure and trap crops, we have thermal requirement models. Here we have a lettuce crop planted in September, October, or November. And what you can see from the galling index, i.e. the severity of the disease caused by the nematode, so the nematode infects the plant and causes these galls. There's an infection in September and in October and November, but it's only in September when there's reproduction. If we look at the thermal requirements of nematodes in lettuces, we can see that the only month when the accumulated temperature has increased for reproduction was in September, not in October or November. These models, when we know them, will allow us to use crops that are of economic interest, that are not of economic interest, I beg your pardon, as trap crops. And the roots are removed, so there's no inoculation. When you use green manure, you have to take into account the incorporation of the crop in the soil, which should we should make sure that the nematodes that are present in the soil won't survive with that. Another method is plant resistance, which operates directly on the nematode. In vegetable crops, we find this in different crops. Some are pending validation, but in some cases they're already commercial or on the point of becoming so. Here you can see nematode reproduction in cultivars. In this case, they're rootstocks of melon and cucumber. With regard to cucumber or rootstocks for watermelon as compared to a watermelon cultivar. So there's an inhibition of the infection with this. What we have seen in tomato cultivars is that when you have successive crops on the same plot, it increases nematode reproduction as compared to what would happen in a susceptible crop, and it can be even 50% over than the reproduction that you would have in a susceptible crop. And so it stops, it's no longer resistant then. When we look at the population dynamic models, you can see the increase in the maximum rate of multiplication and balanced density after every year of planting a resistant crop in the same plot. Here you see the population dynamic in a susceptible crop. So increasingly we're moving towards susceptibility. And this has to be done with correct criteria, otherwise we'll lose this control method. And what criteria is this? Well, crop rotation, which should include host plants or non-host plants like asparagus, trap crops, as I've mentioned, using thermal requirement models, and also resistant crops, poor hosts, and susceptible crops as little as possible, which will lead to increases which will be difficult to manage later. It's important to note that according to the time of year, and so linked to the thermal requirements, some plant species can behave like susceptible or poor hosts or trap crops. So I've mentioned lettuce, but also spinach, chard, and others. So what happens when we use other types of control? Here I've put down chemical and biological control. What type of effect do these have on control of the nematode and the crop that we're going to choose afterwards? When we use control methods that act directly on the multiplication rate, it reduces the maximum multiplication rate, which brings down the residual population that will remain for the next crop. When you use control methods that act on the young nematodes in the soil, you reduce the initial population density, which doesn't change the maximum rate of multiplication, but it does change the balance density. When we use control measures that act on both factors, i.e. on the multiplication rate and on the initial population, for instance, when we use a biological control agent which also induces defense mechanisms in the plant and therefore acts on the viability of the primary inoculum. This brings down the maximum multiplication rate and also brings down the density balance. And in the long term, this is a more sustainable management of the nematode. And this is what we do in what's known in suppressive soils. Lastly, with these population dynamic models and the models that we have to predict the impact, knowing the life cycle of the nematode, with all of this, You can have sustainable management of these nematodes, but all of these aspects must be taken into account, not just one single type of control measure. All of this work has been carried out by a research team comprising various organizations. Here are the names of the people involved. I'd like to thank them. I'd like to thank all of the farmers and the plant protection associations who've helped us in our trials, the bodies and organizations that have given us moral and financial support. And to all of you, thank you very much for listening.
Gracias, Francisco Javier. Ahora tenemos a Josep.
Thank you, Francisco Javier. Now we have the pleasure of hearing Josep Armengola. And though he's already been introduced in the plenary session, I'd just like to stress that he is an agricultural engineer. He works at the Polytechnic University in Valencia. He's also a member of the National Reference Lab for Fungi, and he has had a lengthy career dedicated to studying the fungi that affect plants. He's going to talk about soil-borne fungi, which are more and more important, and especially now when we have less and less chemical options in order to combat these diseases.
Muy bien, buenas tardes. Muchas gracias a la organización.
I'd like to thank the organizers for giving me a chance not to give just one presentation, but actually two. But I feel very at ease in both with both of these topics. And although soil-borne fungi are not one of my main lines of research right now, but they have been something I've worked on very much in the past, and I actually did my thesis on this topic. And therefore, it's an issue I'm very much interested in. I'd like to start off with a very elementary presentation, but I think which is good to have an idea about the main issues where we have to take into account. Very often we have a problem, is that because we have now less tools, we have less ways of controlling the fungi, but also because there are new things that are having an impact on the control of these pathogens, like cultural methods and so on. And although this may sound maybe very familiar or too basic at times, I think it will be useful. Now, let's see. Here we go. Well, diseases caused by soil-borne fungi are diseases which may lead to very high crop losses, and also diseases that may affect the crops right from the initial stages of development right to the moment when the crop comes to its final productive stage in the field. And very often both stages are linked, so we may have an impact of certain fungi in the soil and in the nursery, and then different ones in the actual field. But very often we also find the problems which we have with seedlings are then taken into the field, and then they become a problem in established crops. And here I'd like to talk about that link. There may be, of course, independent problems between the seedlings and the field, but very often there's a link between these. Here we have an example of an issue we had in the Valencia area, about 6 or 8 years ago. This was caused by a fungi, Fusarium solani, the specialized form cucurbitae. It affects pumpkins. It's spread by seeds, and we started seeing an impact in our pumpkin fields. After looking at this, we saw that it came from an infection in the plantlets, because we were having plant material produced when they didn't actually die from in the initial stages. Then they were taken and planted into the field, and that was linked to the situation we were finding. So we had, of course, found the origin of these infections. They were the seedlings that were bringing the fungi into the field, and then the plants were wilting. It wasn't a vascular disease, it was the rot of the roots and the crown. Vegetable crops are very much susceptible to all these problems. I've already mentioned one of the reasons why that's so, because there's a continuous replacement of plant material, which means that very often you have to import seeds which may bring soil pathogens with them, but also because there are new cultivars, new varieties, and we don't really know how susceptible they are to the pathogens we have in the soil, and which may change the soil dynamics. There are also cultural changes, innovations in methods of cultivation, the use of mulches, of plastic mulches in the 90s led to a different degrees of presence of the various fungi. Also because we very often use the same crops in the same place year after year, because the various cooperatives specialize in producing a single kind of crop, and it's difficult for them to have crop rotations because they can't face that constant variability of plants of different species. And also, as we heard at the beginning, a reduction in the availability of plant protective treatments which can be used on the soil. Just as we may have fungi for use on the area parts of the plant, we don't have that many for the soil. I'd also like to talk about some of the characteristics that make it even more difficult to manage soil-borne fungi, and also identify and be able to diagnose which fungus we're up against. And of course, as a national reference lab, I'm in charge of those things. First of all, I'd like to say something which we probably already know quite a bit about, is that the biology and the ecology of the microflora of the soil is extremely complex. It's very hard to be able to have access to the soil and to carry out a good study which allows to know what interactions we have in place there, and to know what the life cycle of the pathogens are. There are interactions between the plants, plant microorganisms, interactions between microorganisms. Also, I'm only going to talk about fungi, but Sorribes just spoke about nematodes, and obviously there are interactions between the fungi and nematodes in the soil, which may lead to it may favor fungal diseases, and we could also talk about that later on. But if we think about interactions between microorganisms, between fungi themselves, and there are many other interactions which we do not know about. And there are also many other factors in the soil that we have to take into account, which do not directly have to do with the ecological aspects of the organisms in the soil, but they also have an impact on the population dynamics. And we also heard about that in the previous presentation. The fact that there is not a high degree of crop rotations, certain cultural measures that inadvertently lead to an increase in some fungi in the soil, because we are favoring the fungi with a higher degree of moisture, with higher temperatures, and so on. And there are also other factors that had an impact on the characteristics of the soil, on the physical and chemical characteristics, texture, pH, the amount of organic matter, the moisture, particularly this area, the bit of soil around the roots. This can have a huge impact on the incidence of fungi diseases, fungal disease, and the impact on the crops. In our lab, we have studied one of these examples that you see here. We're trying to show part of the table. You can't see it very well, but in this case, we have a soil-borne fungus, Monosporascus canumbalus. We've worked very much with it, and the propagula in this case, the survival structures, which are the ones we're interested in, because they're the ones that lead to the primary infections of cucurbits, are these black balls you see there. They're ascospores of the fungi. Those ascospores depend very heavily on the type of soil, not just of crop rotation. Obviously, the more we rotate, we will increase the incidence of propagules, but they also affect the conditions of the soil. So if we quantify the ascospores in different kinds of soil, in this case we worked with soils from Tunisia, but we've also done it in our conditions, local conditions in Valencia, and we saw that some factors, particularly the pH of the soil, have a very distinct impact on the levels of populations. And here we saw that pHs which are near the optimum for the fungus, around 7, will favor an increase in the number of ascospores of the fungus in the soil, and therefore a higher incidence of the disease, which in turn we saw was linked to certain types of soil. Another important aspect to take into account in order to understand how soil-borne fungi are carried out and what disease they lead to is to bear in mind that many of them have survival structures which are survival structures that can last very long, and that makes it even more complicated when we want to establish rotational crop systems and so on, and decide what we're going to plant in an area in the future. Many of these structures are well known, Sclerotia, chlamydospores, but fungi can also survive in leftovers from old crops, and very often it's not enough just to know about these well-known structures in order to understand the dynamics. For example, the Fusarium chlamydospores. Fusarium is a genus that has a lot of representatives. For example, all the vascular diseases which are caused by Fusarium oxysporum, and we know that chlamydospores, which are these resistant structures which are formed in the fungus mycelium, can persist in the ground, and they may affect, for example, cucurbits or tomato in these specialized forms of Fusarium, and they may have survival periods of more than 10 years without planting susceptible crops there. So that knowing and having information about those structures is essential when it comes to determining what crops we're going to plant. Another structure which is interesting is Sclerotia. Sclerotia are structures which act as a survival structure and also as a dispersal structure for soil fungi. If we take soil which is infected with Sclerotia via our tools, for instance, from one place to the other, we can contaminate that area. Some Sclerotia may be very big, like the ones of Sclerotinia sclerotinium. These black spots you see, they're like a kind of peas that appear on the base of this fennel. Shows a great degree of decay there, but there are also smaller ones like the Sclerotia we have in Sclerotium sepiverum in garlic, and this is from Castilla-La Mancha where we're working lately. We see there a lot of impact of this disease, but there's also Sclerotia which are far smaller, the microsclerotia of Microformina, which affect cucurbits and strawberry, and also the ones from Verticillium dahliae, which is responsible for Verticillium wilt of many crops. Another very important aspect to take into account is the interactions of the soil, and we find that very often there are simultaneous infections. There are complex diseases or disease complexes. Very often we speak of one of these, but I think it's better to speak of complexes of diseases. There are various pathogens which impact the crop at the same time, and therefore we have a greater level of affectation of that plant. Sometimes we see an impact on the plant of both fungi and nematodes. Here we have an example of this. This is the cucurbit collapse, which is due to different soil fungi which may interact, or sometimes you have one acting and two, sometimes even three. These fungi lead to a progressive destruction of the root system of the plant. We have Plectosphaera melonis, or Melosporasus canambalus, or Acrolemia vagum. All three of these, due to the destruction of the root system of the plant, may lead to the plant death precisely when there's a need for greater water supply, usually when we are close to the moment we're going to harvest, which is when water is needed by the aerial part of the plant. Another example of the same is the pink root disease of onions caused by Setophoma terrestris. This is a frequent disease which is found in areas where onions and garlics are grown, and this disease is very often aggravated with even greater damage than what is expected, because there are other species of Fusarium in the soil which contribute to increasing the damage to the roots, attacking them at the same time when we already have prior damage by Setophoma terrestris. And the symptoms are typically these pink roots and a progressive loss of the roots, and therefore a lower development or growth of the plant, and a complete loss of production in garlic and onion fields. Now, what are the main kinds of diseases? As I was saying, what are the main kinds of diseases which we may find? The main ones are the diseases that affect the root and the neck, the vascular disease, and sometimes disease of the aerial part due to damage in the crops and the fact that fungi may reach the aerial parts. Here we have some examples. Root and neck can be caused by soil fungi which will attack and destroy the parts of the crop which are in contact with the soil root and the neck. Usually they destroy the root system, or they may cause decays such as the ones caused by Sclerotinia sclerotium in lettuce and many other horticultural crops. We have Phytophthora capsi, the pepper wilt disease, which is probably one of the most important diseases of the peppers, which also affects the roots and the neck and the progressive wilting of the disease. Then we have Rhizotorania solani in beans. Is also one of these important diseases, but can also affect many others. And then we have new diseases which we do not know so much about. Like, for example, in Castilla-La Mancha, in garlic crops, we have found over the last few years a high incidence of Penicillium ali in the field, when this was a problem which was usually found in the post-harvest context. So for some reason, this post-harvest disease is now becoming an important problem in the field, causing the penicillin decay of garlic. So we can never actually say we have managed to control the problem, because these are all very dynamic systems and new diseases come up constantly. Another example of the second kind of diseases, vascular disease. Here we have the verticillium wilt. The vascular diseases are caused by fungi which penetrate via the roots and may go up through the vascular system, damaging it, damaging the transport of nutrients, leading to stunting and wilting of the plant and death of seedlings. I took as an example the verticillium wilt of artichokes, but there are many others in important crops. Some of these vascular diseases can be controlled with using grafting on rootstocks, tolerant rootstocks. This is something which we already mentioned for nematodes. We have another example of vascular disease by Fusarium wilt in melon, but they are also present in many other crops such as tomato, watermelon, and so on. There are also examples of new vascular diseases. Here we have the lettuce trachomatosis, Pythium tracheiphilium. This is a kind of Pythium which has only been detected in Valencia and in Asturias, and this fungus leads to a vascular disease. As you can see, the vascular system is damaged, the fungus penetrates through the roots, and it happens when there's a high degree of moisture particularly in lettuce crops, and it's very hard to control this disease in the soil. And the last example I'm going to give you is the third kind of diseases, the soil fungi, which do not just affect the parts of the plant which are in contact with the ground, with roots and neck, but we must bear in mind that sometimes they can also affect the aerial part of the crops. And here we have an example of this. This happens very often in horticultural crops when there's a lot of rain. This is the presence of Sclerotinia sclerotium in the aerial part of some crops, such as celery and various plant species of the crucifera family, broccoli, cabbage, and so on. And with that, I've come to the end of my presentation. I hope I've given you a general overview of the problems of the type of diseases, because the more we know about these, the better, in order to be able to come up with management systems for our horticultural crops. Thank you very much.
Gracias, Josep. La siguiente ponente es...
Thank you. The next speaker is Soledad Verdejo Lucas, who is a researcher at the IFAPA in plant protection, specializing in nematology, working not only on plant resistance but also biological control of nematodes. And she's carried out research in various research centers in the UK and California. She previously worked in the IRTA, and she's currently working at the La Mojonera Center, where she carries out research on nematodes in horticultural crops. And this afternoon she'll be talking about rootstocks as an alternative in integrated management. Good afternoon, everybody. I'd firstly like to thank the organizing committee for this second edition of the Smart Fruit Congress, for their invitation to talk about rootstocks. This is a technology that's been used in Asia for over a decade now, and it's primarily been used to counteract soil-borne diseases and the damage they cause. The rootstocks give tolerance or resistance to one or several diseases, which allows us to produce cultivars that are valued by consumers in poor or inadequate soils. The rootstocks increase the vigor of the plant and the yield and quality of the fruit. And one of the aspects that affects quality is the uniformity of the product. Some also give tolerance to abiotic stresses, such as low temperatures and salinity. And rootstocks are being used as a technology to control soil-borne diseases considerably more since methyl bromide as a soil fumigant was banned. They're popular among farmers, and those who've adopted this measure like it. There's been a change in their agronomic practices in producing grafted plants. And once most farmers have started that, they don't want to go back to other previous control measures. This is a list of soil-borne pathogens that can be controlled by grafting. The main vegetable crops that are grafted, tomato, aubergine, and pepper as parts of the solanaceous crops, and watermelon, melon, and cucumber in that order, the cucurbit crops. So why are rootstocks used, or why are each of these crops used? In tomato, it's used in Europe and the US primarily to extend the harvesting season. In greenhouses where they're grown in the soil directly, the rootstocks have to be resistant to diseases. The most common use are hybrids of Solanum lycopersicum, which is commercial, and mainly various wild tomatoes, but primarily Abrochitis. And this gives this added vigor, which is one of the characteristics of rootstocks. However, that doesn't mean that there is less fruit production, and that's one of the advantages of rootstocks. Farmers also use these to have two stems on grafted plants. And therefore to have fewer plants. In aubergines, it's mainly used to control soil-borne diseases, Fusarium wilt, and other types of bacterial wilt, root knot nematodes. The most common rootstock used in Japan is Solanum integrifolium, also Cincimbrifolium, and recently Solanum torvum has also been introduced. The drawback of Solanum torvum is seed germination. I mentioned Solanum torvum for aubergines, but it is actually also used for other rootstocks which have difficulties with germination, uniform germination, and scion rootstock compatibility. And also the same is used for tomato. In peppers, it's used mainly to control Phytophthora capsicum and Melodocene incognita. Interspecific hybrids of Capsicum annum are used with different lines of PI, which is what gives the resistance. Rootstocks for peppers have quite limited application and aren't used worldwide. There are various sources of genetic resistance, which extends the possibilities of disease handling by alternating the source of resistance, which doesn't exist with other rootstocks. In Spain, there is a breeding program, an improvement program, to have a pepper that's resistant to salinity. That's going on in Valencia University and others, and also for resistance to M. incognita in Murcia region. In cucurbits, grafting is used in watermelon, melon, and cucumber. There are various rootstocks. Some are used for all of these crops, whereas others are more specific. Generally, they give non-specific protection, but sufficient to counteract a series of pathogens. In watermelon, it's used mainly to control Fusarium oxysporum and Fusarium niveum. You also have Cutea maxima, used mainly in Moscata here in Spain, particularly Laginaria ciceraria. Wild watermelons like Citrulus citroidus as a source of resistance, And also seed companies are now offering Citrullus lanatus, as common watermelon, as a rootstock. In addition to resistance to Fusarium niveum, it has good tolerance to cold, which allows farmers to bring the plantation date forward. One issue that always emerges when we talk about grafting in watermelons is the quality of the fruit. There's been some controversy about this. Studies that have been carried out here in Spain, in Valencia, have shown that if the conditions are controlled correctly, there isn't a negative effect on the quality of the fruit. In melon, the main reason why grafting is carried out is for Fusarium oxysporum melonis, and it's also used to control non-specific pathogens such as Monosporascus cannibalis, which Giuseppe Armengol mentioned, Macrophomina fasolina, and Didymela brayoni. Both melon and watermelon are susceptible to these non-specific pathogens, and it causes a wilting. We have melon cultivars that present resistance to this, and one of the main limitations is rootstock scion compatibility, which means the fruit quality is lower. By way of summary, all rootstocks have the pros and cons, and you have to choose them depending on the pathogen or the limiting factor in the situation, the productive system, whether it's greenhouse or open air, and the environment and the time of year that you want to plant a crop. I'd forgotten to mention cucumber there. Which also causes wilt, Fusarium wilt. And it's used to increase tolerance to the cold, and this is something that's sought particularly in Japan, not so much here in the Mediterranean. It's also used, wild cucumis are also used. Again, in Asia, they're widely used, Cucumis ficifolia, and hybrids of Maxima and Mochata. I'm going to talk now about factors that affect the effectiveness of rootstocks as protection in soil-borne diseases, and I'll use nematodes as the target pathogen here. I have a list of factors on this slide, and I'm going to discuss them briefly now. So the genetic background of the rootstock. Here you have the results of a study looking at different tomato rootstocks compared to resistant susceptible cultivars in accordance with the trials. These were all resistant to the nematode, and this was tested and checked through molecular testing. However, we found variability in the response. Some or most of them were highly resistant, whereas others had intermediate resistance or were entirely susceptible. So the genetic background of the tomato, in this case, but it also works for other rootstocks, has an effect on the effectiveness of this as a control. Variability due to the pathogen also is important, be it its aggressiveness or reproduction. Here we carried out another test looking at various populations of nematodes, you see some rootstocks are highly resistant to all populations, whereas others have little resistance or are susceptible to most of them. So, with the variability of the pathogen, depending on that, certain decisions have to be made. Something else we've tested is the effect of continuous monocropping, how this affects durability of resistance, and if continuous use of rootstocks can give rise to selection of virulent populations. Once the rootstock has been used, farmers usually want to continue with the same. And here we define virulence as the pathogen's capacity to significantly reproduce on resistant plants. This is an overview of the experiment we carried out. 3 cycles of continuous cropping and soil that was infested with an avirulent population. This graph shows the populations that we find every year at the end of each of the crop cycles in the various materials. And as you can see, there's progressive accumulation of the inoculum year after year. And because of the effect of the genotype that I mentioned earlier, you can see also that this accumulation changes according to the material being used. We can see this in the number of nematodes in the soil, as well as the severity of the disease and egg production. And this shows how it affects the virulence of the disease. In the first year, all plants behave as highly resistant. This is maintained after 1 crop cycle. In the second year, the resistance has dropped and it's now intermediate resistant rootstock, and in year 3 it's completely susceptible. So it's the same, it would be the same as using a susceptible crop. So continuous use of rootstocks can give rise to rapid selection of virulent populations in quite a short period of time. Now, then we went to the field to verify the situation with the existence of virulent populations in the field. This was a study carried out in Almeria of 17 nematode populations in Almeria. 9 of those populations had total virulence or partial virulence. And this indicates the future of rootstocks if, in such a short period of time, just a few years, they've selected virulent populations. One factor that we think contributes to this, the high number of virulent populations that we find so quickly, is the fact that they're not completely resistant, but have partial resistance. And because they have partial resistance, the virulence selection process speeds up. Another point I wanted to touch upon is from the response to the target pathogen, how it responds to other pathogens in the soil. In cucurbits, we've seen that rootstocks are used to combat Fusarium wilt mainly, and they're very effective against different types of Fusarium. However, with melanogen nematodes, they're extremely susceptible, and there are high population increases after use of these rootstocks. This is a comparison of multiplication of rootstocks and multiplication with watermelon cultivars. You can see then that the difference are visible in both of the graphs, with watermelon as a host, as Javier Sorribes has mentioned. There are low population increases, poor host status, compared to good host status. So also tolerance and resistance. Tolerance is very good for the crop that it's being used in, but it has to be used carefully, because when it comes to nematodes, with a severity of galls, as you can see here, the rootstock produces more than an ungrafted plant or a plant grafted on another type of rootstock. So here you can see that the choice of the rootstock or the rootstock-scion combination is very important for different types of problem. If there are very high population levels at the end, it can affect the next crop in the crop rotation. And just to recap then, grafting is a very valuable tool in integrated disease management, but it must be used as part of integrated management alongside other tools and with in-depth knowledge of the system where it's going to be used, to preserve the durability of the resistance and avoid selection of virulent populations that would make it useless. I think in the future, this rootstock-scion combination will be increasingly important. It already is today, both to deal with incompatibility problems, and it should also be based on the grower's needs, the farmer's needs, rather than nursery preferences. Rootstocks are used that have good germination and compatibility that don't necessarily meet the farmer's needs as exactly. So improvement programs need to select material that responds to certain criteria, and there should be a la carte rootstocks, if you like, to meet specific needs. Thank you very much.
Gracias, Soledad.
Thank you very much, Soledad. Thank you for sticking to time, and thank you for that extremely interesting presentation. Our next speaker is Miguel Talavera. Miguel Talavera has a doctorate in biological sciences. He conducted his thesis on nematode survival strategies. He has undergone a series of postdoctoral study programs in the UK and in Japan, working on the biological control of nematodes, and he's right now head researcher in IFAPA in Granada. Whenever you wish, you have the floor to talk about biological control of nematodes. Good afternoon to all of you. I'd also like to thank the organizers of Smart Fruit for having invited me to participate in this roundtable. Now, my presentation in this case is going to talk about the different methods we have for controlling nematodes in soil. It is based on the work we've conducted in the field, in our experimental fields, and also working with companies, and it is going to be very practical in nature about how we've been able to control nematodes over the last 10 years. First of all, I'd like to give you an idea of where we work. This corresponds to the area of protected crops in Andalusia. It's a quite large extension, almost 45,000 hectares distributed amongst these 37,000 along Almeria and the coast, and the 17,000 hectares in Huelva on strawberry production, and also a small production we have for cut flowers between Cádiz and Seville. In the slide you can see what we produce, the surface dedicated to each of these, and on a yearly basis this means that farmers read about 2.5 billion euros per year. That's about a third of the agricultural revenues in Andalusia. So it's really important. Secondly, we had to have a look on whether they were affected by nematodes or not. So we did that taking a twofold perspective into account. First of all, we conducted a survey to the specialists who were in charge of the health of crops in the fields. We asked them whether they perceived that there were problems with nematodes. And here you can see that depending on their answers, and this was done just in Almeria, that we don't have the data for Huelva yet. This is just for Almeria, but approximately about 20% of the plots had problems with nematodes. 20% of the greenhouses had problems with nematodes, and we have the data divided here between the two areas because they have different cultural methods, and that had a certain impact on how to control these diseases. But I'm not going to go into those details. I'm just making an average of both areas. So about 20% of affectation. As you know, the nematodes are distributed in round areas within the field. So we asked them what surface of the field they considered to be affected, and there's another result of about 20%. So if we bring that together, we see that about 2% of the surface dedicated to intensive horticultural crops in Almeria is infested by nematodes. When we asked them about the losses, it's about 30% of the yield losses, and which is 3% of production. When it comes to distribution of species, obviously we conducted a sampling of the plots to make sure that the information we had received via the survey was right, and also to identify the species which were present in the greenhouses in Almeria. In our sampling, we found a percentage of plots affected a bit higher than what the expert had expected, which is quite logical, because unless you have clear galling symptoms, you may not perceive that there are nematodes there. Regarding the species present, we obtained these results, which is interesting due to the different distribution we have in those two areas. In the Levante area, where there is a tomato monoculture, the most prevalent species was Meladogine javanica, which is more virulent and reproduces better in tomatoes. However, in the Ponente area, where there is a crop rotation between nightshade family plants and cucurbits, there is a more equal presence of the various species, but there we had a predominance of Meloidogine incognita, followed by javanica, because many of the varieties which are planted there are resistant to Meloidogine javanica. Now, so what do the agricultural technicians do about these issues? This is a survey conducted in 2012, and at that time there was still 78% of the agricultural technicians that used fumigant nematicides when they had problems with nematodes. 60% used non-fumigant nematicides, and of course this doesn't add up to 100% because very often they applied various methods. Resistance and grafting 42%, soil solarization 38%, biofumigation only 6%, and other products biostimulants, mycorrhizae, plant extracts, and so on. There are many, many other products which are found on the market that quickly disappear, are used by 38%. Now, I always show this slide because when it comes to the biological control of nematodes, it is something which is different when it compared to normal IPM caused by other pathogens. This shows how we can improve our control methods or control strategy as we know more and more about the scientific aspects. First of all, we use indiscriminate treatment, use of soil fumigants without knowing what we have present in the soil, just as a preventive measure. Then, as we know more about the pest or the disease, we start going through different stages till we reach the integrated pest management. Well, of course, there we're not going to stop because this is a dynamic control. We're going to come up against different problems, but just has to show. Goes to show how we adapt our strategies according to scientific knowledge. If most farmers in this area still use fumigant nematicides, it's maybe because we haven't given them the scientific knowledge which they need in order to conduct integrated pest management measures. And I'd like to talk more about that later on if we have time for it. Now, how do we manage nematodes? We do it take into account this graph. If we take into account the losses in the crop, when there is an increasing density of nematodes, we always have a graph of this kind. At some point, which is what is the tolerance limit, there are visible losses, and then there are other thresholds, which are the damage thresholds, which will depend on the cost of the control measures. These various thresholds and so on are now being implemented in the various crops. Javier showed us some of them, and they are a fundamental tool in order to be able to control nematode diseases in a rational manner. Now, what is an integrated nematode management system? As we saw before, if we control the initial density of nematodes when it comes to transplant, we will reduce losses. That is a preventive strategy in our crops, but we also have to take into account other aspects in our integrated approach. For example, if we have an agricultural system with a succession of crops, we should also think that, bear in mind that the nematode population is increasing and the inoculum is increasing. So there are other measures which decrease the nematode reproduction factor, and that should also be part of our integrated nematode management system. And then there are other measures which do not neither reduce the nematode charge nor the reproduction rate, but they compensate for the damage done by nematodes into plants. And that can be very useful at some point in time, as long as we take into account that at the end of our crop production, we're going to have a high level of nematodes because we haven't reduced their reproduction rates. Here we have a summary of all the methods, control methods, which are used for nematodes. Some are widely used in our area, some are not used at all, or some are simply not applicable. And we're going to take a look at what the advantages and limitations are for each of these methods, and which is what we've seen over the last 10 years in which we've been working in the field. First of all, we have agrochemicals. This is a traditional method in our struggle against nematodes. Of course, they've been widely tested in many kinds of conditions. They are commercially, or they were commercially available, most of them, and the efficacy was well contrasted in various kinds of environments. Obviously, there are certain limitations which are very serious, and that's why many of them are now forbidden or have a very restricted use. Basically, health and environmental risks. In this table here, we can see what agrochemical products are authorized right now within Annex 1 of the European Directive to use for nematodes. Now, that doesn't mean that we can use all of them for all crops. As you know, there are limitations according to the crop, and very often we just can't use them freely. First of all, we have 1,3-dichloropropene, which is not included in Annex 1. It can't be used, but we have provisional exemptions right now, and you can still use it in the field. The rest of them are included in Annex 1. You have their trade names, but there is a problem. Before, we had far more chemical products available to combat nematodes, but now we don't have that many. So, taking that into account, we decided to develop alternatives to methyl bromide. We tried out chemical alternatives in these initial projects, but they also compared them to other alternatives, physical and biological and cultural methods. And I'll show you the results of some of these tests we carried out, particularly with strawberries, where we compared the efficacy of these chemical products when it came to reducing nematode populations. The results are based at least on 3 repetitions in the same plot over a 3-year period, and we have corrected for the mortality nematodes have in normal conditions without treatment in the period prior to the treatment and the one after the treatment. So in general terms, what we see is that there is a group of agrochemical agents have an efficacy rate of about 80 or 90% in a reduction of nematodes. These are fumigants. You have methyl iodide is probably never going to be authorized again. It was authorized in the United States for a while, but then it was withdrawn due to its high toxicity. But methyl sulfide disulfide may be authorized, and in a couple of years it may be on the market. Then there's a second group of substances, such as methyl sodium and a whole series of chemical products where the efficacy is not very satisfactory. This is for meloidogeny, and this is for pratylenchus, which is another pathogen we find with strawberry crops. The products are similar, and the efficacy is a bit lower for pratylenchus, basically because this pathogen lives within the roots and the product does not reach. Now, the second kind of control method is resistance and grafting, because that should be very effective. If you have a resistant cultivar, in theory, we should have an efficacy of 100%. It's very easy to use if the cultivar is resistant, but there are certain limitations also. The ones Soledad Verdejo or Chavez also mentioned before, virulence, and in the case of resistance in tomato plants, this resistance is not effective when the soil temperature is very high, which is what happens very often in greenhouses in Ana Maria. Here we have a graph which you've already seen before, which shows that practically 50% of the populations which we sampled in Ana Maria already had symptoms of virulence to this nematode. And in Japan, when I was there, 80% of greenhouses were virulent. So if you don't manage those resistances appropriately as a control measure, we may find that the resistance is no longer a suitable control method. Solarization, we all know how it works. You cover the soil with a plastic film so that, well, you wet it first, of course, and so that solar rays increase the temperature, and that should kill or at least inactivate the nematodes. It's very good for other pathogens also, particularly fungi. But in the case of nematodes, in addition to needing a higher temperature, we also have the problem of the mobility, because they can move, of course. And the nematodes which are in the deeper down in the soil are not affected by the temperatures because it's not high enough, and they can quickly move up again and reinfect our crops. Biofumigation. Biofumigation, as you also know, it means adding fresh organic matter to the soil, wet it, seal it with plastic so that the gases created during fermentation can kill off a great number of pathogens. It improves the structure of soil. It uses local resources in most cases. However, it does have a series of disadvantages. The product use is very variable. We have inconsistent results, and there may also be a scarcity of organic matter if we start having widespread use of this method. Biological control, I wasn't going to speak about this, but I'm sorry. But in the case of nematodes, it's something which is being developed now. Very often in our preliminary test in pots or in stable soils, we find a very high efficacy, and in theory there are various biological agents which are active, but the problem is, on the one hand, the commercial production of them, and secondly, the establishment of these in natural soil conditions where they have to compete with the microflora and the microfauna of the soil. In general terms, the studies we carried out in the field with biological control agents have shown quite low efficacy rate, and that's important because they may work very well as an additional help, but if there's a very high density of nematodes originally, then in themselves they're not enough to control that. I also like to show this slide because we sampled biological control agents in Almeria. We found a lot of fungi and parasites of nematode eggs, but the rate of parasitism was very low. There's another agent, which is Posterior penetrans, which was not present in this area, but we do find that in other parts of Spain. So maybe the production methods in the greenhouses in Almeria does not allow for the establishment of the populations of these fungi so that they can be effective. That doesn't mean, of course, that if we changed our cultivation methods, that would change. There are other control measures such as crop rotation, land fallow, using allelopathic plants, which later can be tilled into the ground as green manure. But most of the intensive horticultural methods cannot use it because of the low financial margins. But there are different methods, some of which may be able to be used at some point within the greenhouses. And finally, our goal will be to develop databases and control systems so that we can transfer this information to the farmers and to the experts in the area so that they can have information in order to make suitable decisions depending on the density of nematodes they find in their soil. Now, this is just a summary of what I've already said, but I'll leave it there for you to take a look at your leisure. Thank you very much.
Gracias, Miguel. Y efectivamente.
Thank you, Miguel. And yes, I'm sorry, I made a mistake in my presentation. You talked about integrated management and not biological control. So we're going to have our Q&A session, and I hope you'll all participate.
Por favor, lo que sí que pediría era que las intervenciones sean cortas.
Please do keep your questions or comments brief. The idea is to have a bit of a debate. Antonia Montserrat, I'll be very brief, and I have a question for Miguel Talavera and Mr. Sorribas. Recently, we've seen an increase in nematode populations in open air and extensive crops and brassicas, for example. How can we react to that to halter this growth? And also, we're moving from very susceptible soil to suppressive soil. Broadly speaking, if you can do crop rotation, we have very specific nematodes. It's probably what you've got is a parasite of broccoli and so on, and it can't be rotated with other crops or always. But if crop rotation is possible, then you should use integrated management of this type. Is crop rotation possible for broccoli or monocrop that's planted year after year? They're rotated. In the case of pteroderas, what's also used are trap crops, depending on the extent of emergence from the roots. Some need the host plant to be activated, and there are some types of plants. Some companies distribute biocide plants, as they're known, and they use this green manure. And when there's a certain number of individuals in the root, they can change the fertility of the soil and prevent the nematode developing. But it's important to really see the emergence of youngs and penetration of the roots. If that's effective, it could be a good solution. And the second, from a conductive soil to a suppressive soil, to move from that requires a greater depth work, and it's a more of a long-term project, and that's increasing the biological fertility of the soil. What we have seen, what Miguel has mentioned, is percentages of parasitism that we've seen here in Catalonia, and from that we've seen that in soils where there was greater enzyme activity, kiniolytic activity, and so on. By analyzing the composition of the microbial flora, you can see that where there's percentages of in certain areas there's very high population of parasitism, and with 3-year studies on these soils, we've seen that the nematode is present, but the multiplication is relatively low, and there are almost no symptoms in the crop, so it is sustainable, and that's what we're working on at the moment. But it's a long-term project to improve soil fertilization with organic matter and so on. Going into a bit more depth on this, I think it's important to identify the nematode. From what you've said, it might seem to be a heteroderus, but you have to know that specifically because part of these trap crops and so on differ according to the species, and there are various crops that where these are being used for rotation for beetroot. If you know which species is concerned specifically, then you can use these biocides, which are often wild radishes and so on. Yes, picking up on this, I want to ask another question. You've talked about changes in crop systems and company specialization. Companies are increasingly specialized, and work with 2, 3, or 4 species. And what you have is that it's a repetition of the species year after year of the same on the same plot. But they say, oh, we'll re-rotate the crops. Do you consider that to be crop rotation, or is it monocrop if it's different species? It depends on the pathogen. If it's Melodynia, it's probably a monocrop because both in Solanaceous and cucurbit and other families are hosts of Melodynia. With a more specific pathogen, it might be different. We have similar cases, an increase in problems caused by sclerotonium in many crops. But all of these crops are hosts. You can change the lettuces for broccoli and so on, but they're all susceptible hosts. So you have an increase of sclerosis in the soil. And whereas this wasn't a problem in the past, it is very much now. So it's not rotation because it's a pathogen that affects different hosts of different species. Are there any more questions? I'm from a plant protection service. I'd like Dr. Talavera to experience results of of their experiences. Well, in solanization, we have ruled it out as an effective control measure in greenhouses for 2 reasons. Because we didn't have sufficiently high temperatures. It's strange, but you get higher temperatures in the soil in open air than in greenhouses in certain areas because it interferes with the crop cycle. At higher temperature, salinization might be more effective, and also because, as we've seen, the residual population of nematodes that survive easily reinfects the crop when it's established. In any case, we do use it in combination with nematicides, and particularly with biofumigation. Some people call this biosolarization, but it's much more effective when you use it with solarization. With the gases under plastics create higher temperatures. And one of the measures, which I forgot to mention, is there's a comparison between different agrochemicals. But if you look at the presentation afterwards, you can see that we have that biosolarization is around 78% effective. So it's comparable to many of the nematicide agrochemicals that are on the market. So we use it for alone. We don't recommend it for nematodes, but it does work very well for other pathogens like Fusarium and Vertinium in greenhouses in Almeria. I have a question for Soledad. You mentioned the problems in peppers of Phytophthora, which meant that much of agricultural land was unviable for planting peppers. Do you think that greenhouses that are devoted exclusively to peppers, as you get in Murcia, the increase of nematode populations can mean that peppers can't be grown in that area? Pepper is quite a tolerant plant, in the sense that the nematode causes little deformation. So you can have higher populations than with other crops and maintain crop production. It's perhaps a paradox, but on the one hand you've got production, and on the other the nematode. But farmers obviously are only interested logically in production, whereas we're more interested in a nematode. So if it doesn't affect production, then measures usually aren't taken. But it can affect them, and when there are very high levels, measures are taken at the start. These might be more sporadic measures, because at the end of the pepper cultivation, you have the summer. So there's a drop in the population, as Javier mentioned, because of natural mortality. So in this same production system, at the start, when you get to the start of it again, the population is low. And fortunately, it drops and then it rises again. But the good thing is that at the start, the population is low, and that maintains the system. We have other much more aggressive pathogens that kill the plant. The nematode just weakens it and can coexist. Are there any more questions?
Yo con Sorriba no sé si con tanta gráfica me he perdido.
I don't know if I missed something with all of the graphs that Mr. Sorribas gave us. One thing that I've understood is that when population levels are low, the multiplication rate of nematodes is extremely fast and gives rise to females, whereas if the levels are high, this is reduced and it gives rise to males. So these are techniques that can reduce nematodes in soil by 80%. And is this really effective in the development of the crop? Yes, because the crop production is linked to the levels that you have at the start, at the moment of plantation. If you can reduce the initial population level, the crop losses will automatically be reduced. The fact that the population increases or the maximum multiplication rate increases, that means that the population that has remained after the control measure will multiply n times. The same as with the low population density. Do you get it? So it's proportional to the initial population, right? And what drops is the multiplication rate. That doesn't mean, though, that the end population will not be far less. When you have a population density, you get this density balance, which is what the plant can withstand. Does that answer your question? Microphone for the speaker, please. Interpreter can't hear the intervention. No, it's not that it's fast. It has to do with the intensity. If you look at that graph of the initial and the final population, it's proportional to the initial population. If you have a low initial population, it'll be proportionally low later, but it will multiply. When the population is very high, it means that that population that you have at the start is 1.5 times or 1 time or half a time or 0.9 times what you had at the start, because there is competition. The population at the end of the cultivation is lower than what it was at the start, and that regulates the end population that you have just by a survival mechanism. The nematode has mechanisms to survive and remain in the land that it has colonized. So that's how it works, really. Mathematically, I don't know if it's expressed this way, but it's a multiplication rate. So there can be negative multiplication rates when the initial population is very high, competition is very strong, and a high percentage of the population isn't successful in infecting and reproducing. The nematode is regulated according to the competition there is. There's not an exact number of eggs that the female will lay. I said from 220 to 2,000, but a female can lay 10 eggs if there's a lot of competition around, and the young nematodes will emerge as males. But as Giuseppe was saying, if it's with the sclerosis and the sconiaspores and so on, these are mechanisms that they seek to survive. And as they have such a high range of hosts, it doesn't have to stay for 20-odd years in the soil. If a plant comes up, it can infect it. I have a question for Miguel. You mentioned there are increasingly fewer products and increasingly fewer employment possibilities or use possibilities. Do you think with current doses this is controlled? Yes. It all comes down to effectiveness. We're talking about effectiveness in reducing populations. And there are extreme cases that we've found with very high densities of nematodes in the soil, which is when we needed 90% effectiveness. There have been few and far between of these. Usually we get by with lower effectiveness. But what I think is clear is that managing nematode diseases requires integrated control and knowledge that has to be transferred to technical advisors and farmers because they have to make decisions about it. You can't just say, oh, I'll use a disinfectant and I'll forget this until next year. You have to make decisions, and that's what we're working on. And Giuseppe, I want to mention something. You said that changes in production systems, for example, in very classic species like spinach and chard, with baby leaf, which has nothing to do with conventional spinach or chard, entails new cultivation systems where fungus problems are far greater. Should we set down research strategies to change cultivation systems and to minimize these soil-borne fungi? It's true that cultivation changes usually mean that we have to understand how these affect diseases and pathogen populations. I don't know whether we should be doing this research before that happens or perhaps afterwards. Usually we research things after changes in cultivation, and we understand the role that the changes have played in incidence of pathogens or specific diseases. I think trying to predict what will happen is difficult. It's not really something you can research beforehand. But if the outcome of a change is greater, more infections, more fungus-borne infections or something else, then we have to take measures when we understand how the change has affected the pathogen dynamic. And there are many examples in the soil. For example, drip irrigation, having the water very close by and very constant to the roots and the necks of plants, bringing in other types of measures have also favored rot, also changes in density. Many cabbages and cauliflowers are cultivated more densely, but that means that in the soil there's less ventilation and an increased incidence of fungus like sclerotinia, or it can cause high humidity in the aerial parts of the plants. So usually we research things after they've happened. You can't really predict what will happen. But when you have seen the change, we try to understand how changes favor pathogens and then take measures to avoid it. So if there are no more questions, I think by way of conclusion, I can say that soil is a living system. It's vital to have knowledge of it and to know the health and nutritional aspects, among others, to implement specific strategies to control problems. People from Murcia will also talk about water shortages, but probably in the future we're going to see land shortages. Thank you very much.